Now I am going to explain the general characteristics of Cyanobacteria. It is a group which was placed initially in the algae as a schizophyceae or mixophyceae, but the features are closely resemble to that of bacteria. Hence, now it is considered to be a cyanobacteria. The organisms in this group are photosynthetic in nature and they are all prokaryotic microorganisms. Coming to the introduction, in this presentation I am going to explain the introduction, general characters and the significance of cyanobacteria. They are included under the division Cyanophyta. The synonyms for this Cyanophyceae are schizophyceae, mixophyceae or phycochromophyceae. As mentioned, these bacteria are formerly known as blue-green algae because of the color that they appear. The color is due to the presence of certain pigments. Cyano has a meaning of blue. Bacteria here shows that these organisms are prokaryotic rather than the eukaryotic algae. And the fossils of these organisms are termed to be stromatolites. These are the first organisms that have carried the photosynthetic process on the earth 3.5 billion years ago, and these organisms helped and created the earth's environment to be oxic. So oxygen generating process was first seen in this group of bacteria or the organisms, and these are the first organisms which have the two photosystems that could lead to the production of organic material and that could release the oxygen as a byproduct. And these organisms also have the important feature of fixing the atmospheric nitrogen in a specialized structures which are called as heterosis. So the organisms in this group have two features of fixing the carbon dioxide as well as the fixing the atmospheric nitrogen. Coming to the habitat, they are almost present in every environment. They are present in the aquatic form and terrestrial, freshwater and marine, and even in the soil and even the bare rocks. They are present in the planktonic cells that are suspended in the water forms, and they are present either in the form of phototrophic biofilms in freshwater and in marine water. Examples of organisms which live in the freshwater or Nasdaq, Anabina, Rivillaria, and the examples of organisms which live in the marine habitat is Dermocarpa, Trichodesmium, and few other organisms they live in association with other life forms like fungi in the form of lichens or in the plants and protists and sponges in a mutual beneficial relationship. Example for such a symbiotic relationship is Anabina psychoididae, which is present in the coralloid roots of cycas. These aquatic cyanobacteria, they are best known for the formation of blooms in the freshwater and the marine water, and they appear like a blue-green paint or a scum in the water bodies. Another one is these organisms precipitate the minerals such as calcium and magnesium and could lead to the formation of a travertine, which is a form of limestone. Example for this is schizothrix. Calcium carbonate is deposited by schizothrix, Lingbaia and Rivillaria species. This is about the habitat. That means they can live in the freshwater, marine water and in the terrestrial. Moving into the morphology of these organisms, The vegetative body of Cyanobacteria is called as a thallus. The size ranges in between 1 to 10 micrometers in diameter, and based on the organization of this thallus, there are three types, that is a unicellular form, colonial form, and filamentous form. So in the unicellular, that means they are single, they are solitary and covered with mucilaginous envelope. Example, Senicoccus, Crucoccus, Gleocapsa, Xenococcus. So this is how the unicellular form will be appear. And the colonial forms, here the cells remain together after the division. They fail to segregate after the division and form a colonies. So all these colonies again they are covered with a mucilaginous sheath. Example of such colonial form is observed in Microcystis, Ephenocapsa, Dermocarpa and nostoc. Filamentous nature, the cell division occurs in a single plane, in a single direction, and form a thread-like structure. That's what we call as a trichome. Trichome is a row of cells in a filament, and the trichome with the sheath, the trichome with the enclosing sheath is called as a filament. The trichome which is covered with a sheath is called as a filament. So the filaments, they may be arranged in a form of a single row in the uniseriate form. Example is spirulina, nostoc, or else if they are multisteriate, there may be a false branching observed in the cytonema. Mucilaginous sheath is present in all the forms of either unicellular, colonial and filamentous forms. The mucilaginous sheath generally gives protection from the desiccation and even helpful for the gliding movement. The color of the sheath even indicates the environment in which they are present. If they are in red color, that means the environment is acidic in nature. If they are in blue color, the basic in nature environment. And if it is in yellow or brown color, that means the environmental conditions in which they live contains the high salt concentrations, etc. On the whole, the Cyanobacteria, the organization of thallus is in three forms, either unicellular, colonial and filamentous form. If they are filamentous, they may be branching. It is a pseudo branching generally observed in the cytonema. There is a differentiation in the filamentous cells in the form of vegetative cells, akineids and heterosis. Heterosis are the structures, they are thick walled structures and they are in large size and compared to the vegetative cell, and at the point of contact with the filamentous cell there is a pore where the heterosis contacts with the filament, that there is a pore. So the pore with the polar nodules are present in this heterosis. Based on the position of the heterosis in the trichome, they are terminal, they are located at a terminal portion of the filament. Example, Rivularia. Intercalary, if the heterosis are located in the central region of the filament, then that position is called as intercalary. And example in nostoc. Pedicellate, if the heterosis are present and located in the branches, particularly in the pseudo branches where it is here in the picture. So cytonema. Filamentous cells possess the differentiation of vegetative cells and heterosis. There is a structure called akineids. Akineids are the spores produced in unfavorable conditions, which are the resistant structures. When the favorable conditions are, these akineids germinate into a vegetative cell. At the point of contact to the filament, there is a pore in the heterosis. So the intercalary heterosis are bipored, that means they have two pores, whereas in the terminal heterosis it has only one pore at the point of contact with the filament. The functions of heterosis include, they are the storehouse of the reserve food material and they help in the nitrogen fixation process they contain an enzyme nitrogenase which could help in the fixation of nitrogen. The heterosis help to break the trichome into fragments in the vegetative reproduction. Cell structure. Cell structure is of prokaryotic nature. General features of prokaryotic cell are applicable here, like having the incipient nucleus, that means the nucleus without a nuclear membrane. This structure shows that there are three membranes, that is a sheath, eukaryotic sheath, it is a cell wall, that is a peptidoglycan layer, and they are having the outer membrane. The prokaryotes are of gram negative in nature and contains the plasma membrane. The three membranes are present in the form of a sheath, cell wall and plasma membrane. Here sheath increases the water retention and water absorbing capacity of the cell and helpful to survive in the dry habitats. If the sheath is colored, even that help the cell to protect from the radiation. The cell wall is gram negative, so it contains an outer membrane, and plasma membrane, it is the innermost layer that surrounds the cytoplasm, that's surrounding the cell, which is made up of with the lipoproteins that lack the sterols. And if there are inner foldings of the cytoplasmic membrane, they are called as mesosomes. And cytoplasm lacks a membrane bound organelles like chloroplast, mitochondria, endoplasmic reticulum and Golgi complex. It contains two regions, that is the outer chromoplasm and central centroplasm. The chromoplasm is rich with a photosynthetic apparatus and cyanophacian granules, so it is colored, whereas the centroplasm, it contains the DNA, that is a genetic material. Along with this, the micro compartments like structures that are carboxysomes are present. They are helpful for the fixation of carbon dioxide from the atmosphere, and the carboxysomes, they have the enzyme, that is a ribisco. This is the enzyme involved in the fixation of carbon dioxide in the Calvin cycle. Another is thylakoids. Thylakoids are the separate structures that are present in the cytoplasm, distributed in the cytoplasm, which contain the photosynthetic pigments, which are the chlorophylls, carotenoids and phycobilins. The cytoplasm of the cyanobacteria, they lack the membrane bound organelles and contains the thylakoids. The pigments that are present in the cyanobacteria include chlorophylls, carotenoids and phycobilin proteins. Chlorophylls are the green color pigments. Only chlorophyll A is present in the cyanobacteria. It is the major photosynthetic pigment in the cyanobacteria. Carotenoids are two types, carotins and xanthophylls. Carotins are the orange color pigments. The carotins present in the cyanobacteria are beta carotene and flavosin. Xanthophylls are the yellow or the brown color pigments. Examples of such xanthophylls include mixoxanthin and mixoxanthol. Another group of pigments that are present in the cytobacteria are the phycobilins. They are either blue or red colored pigments, either phycocyanin or phycoerythrin. These pigments serve as an accessory pigments, help to trap the light energy of lower wavelengths, which could not be absorbed by the chlorophyll and pass them to the chlorophyll A. So, that is the role of pigments. Other features include the reserve food materials. The reserve food materials is in the form of a starch. The starch here it is termed to be a cyanophacian starch, and if it is a protein then they are called as cyanophacian granules, and if it is a lipids they are called as oil droplets. When it comes to a locomotion, the unicellular bacteria are non-motile, but the trichome formers, they can move by means of a gliding, and those motile filaments of cyanobacteria are called as a hormogonia. Flagella are absent as they are non-motile. The nutrition here, that is a photolithotrophic mode. It contains two photosystems. Photosystem 2, here it splits the water, serves as an electron donor and release the oxygen. Carbon dioxide utilized as a carbon source that could be converted into a starch material. Photosynthetic apparatus, that includes the pigments, reaction center and the electron transport system. All these three are parts of the photosynthetic apparatus. That photosynthetic apparatus is present in the thylakoids. These thylakoids are again distributed in the cytoplasm. As mentioned in the previous slide, that is there are specialized structures which are helpful for the fixation of nitrogen. They are called as a heterosis. Heterosis are the structures, they contain the nitrogenous enzyme and fix the atmospheric nitrogen, and they convert that into a ammonia, and this ammonia will be transported to the vegetative cells. Reproduction. Cyanobacteria reproduce by vegetative and asexual methods. The vegetative modes of reproduction include cell division, that is observed in the crucocales. Fragmentation, here the fragment breaks up and the new fragments will be produced, which again form a new vegetative thallus, that is called as the fragmentation. And the hormogones, the trichomes are fragmented and produce the motile structures which are enclosed in a sheath, and example is nostochales. Achenates, these are the resting spores, actually these are the resting spores or orthospores, which are resistant structures, thick walled and accumulate the food. The reproduction in the cyanobacteria is by means of either vegetative mode of reproduction and the asexual reproduction. Vegetative mode of reproduction is by cell division. It is the common method of cell division which is observed in the crucocales. Here the cell becomes constricted in the middle and it undergoes division and produces two separate cells. Whereas in the fragmentation of the colony or the filaments, here the colonies are fragmented and the filaments are fragmented into small bits. Now each fragment or the bit develop into a colony by the division of cells in the different planes. Hormogones, here the trichomes are fragmented and hormogones are produced. These hormogones are the motile structures present inside the sheath. Example for such kind of reproduction is in nostochales. Another one is achenates. The filamentous bacteria, they accumulate the much of the food and become thick walled, and such hard resistant structures are called as achenates, or the resting spores are orthospores. The second mode of reproduction is asexual reproduction. It takes place by means of the production of spores. Two types of spores are produced. One is called as endospores, another one is called as an exospores. Endospores are the structures that are produced inside the cell. Here during the production of these spores, the cytoplasm of the cell here becomes cleaved into several bits. These bits later converted into endospores and they are liberated. Such kind of reproduction formation of endospores is observed in the dermocarpa. Exospores, these are produced outside the cell by means of a constriction, and here example is a chemiesiphon, which is epiphytic on the oedogonium. Now moving to the economic importance, cyanobacteria, they serve as a food for the fish in the aquatic forms. So example for the such organisms like Oscillatoria microcystlingbaia, they furnish the food for the fish in the aquatic life, aquatic animals, and some species with a gelatinous layer like nostoc, they can be used in the soups as a food. It can be utilized. They can be utilized as a food. Another one is they are diazotrophs. That means have the ability to fix the nitrogen. So by means of this process, it improves the nitrogen content of the soil, helpful to improve the soil fertility. So now these cyanobacteria are employed as a biofertilizers to improve the nitrogen fixation in the crops. These bacteria prevent the soil erosion. How means they aggregate the loose unbound soil particles so that the texture of the soil can be improved. Like that, it can prevent the soil erosion. Another important one is spirulina. Spirulina is a high content of proteins, and not only the protein, it contains many minerals like calcium, magnesium, iron, zinc, and vitamin B12. So at present it is used as a source of food and feed, and it is used as a single cell protein. And another feature of this cyanobacteria is reclamation of the alkaline soils. It was found that some cyanobacteria, they form a thick stratum on the surface of the saline soils that could help to decrease the pH to a neutrality, helpful for the growth of the plants. Cyanobacteria even they cause diseases if the water is contaminated with with some organisms consumed, then it could lead to diseases like liver, heart and lung diseases in the animals, particularly in the cattle, which consume the contaminated water. Of the species like Microcystis and Anabina, they produce toxins. Anabina, it produces enatoxin, and Microcystis, it produces a neurotoxin. Even the water contaminated with the oscillatoria, if the water contaminated with the oscillatoria species is consumed, it could produce the symptoms of itching, conjunctivitis and blocking nostril passages and asthma. So some of the species of the cyanobacteria cause diseases in the cattle. Summarizing the today's class is about the cyanobacteria. These are the first group of organisms released the oxygen on the its atmosphere, and these are the group of organisms having the ability to fix the carbon dioxide and the nitrogen. They are prokaryotic in nature. Photosynthetic product is a starch that is stored in the form of a cyanophacian starch, and the important pigments include the chlorophylls and phycocyanin. The thallus organization may be either unicellular, colonial and filamentous. The thallus is differentiated into vegetative cells, heterosis and the echinates. Heterosis are the thick walled structures differentiating from the vegetative cells and are able to fix the atmospheric nitrogen into ammonia. These organisms, they do not possess any reproductive structures, simply they undergo either binary fission or fragmentation or by means of production of asexual spores. So these are the points to be remembered in the cyanobacteria. Thank you.